Name is Alec Mayfield, and I'm a graduate student at Auburn University. And today I'm going to be talking a little bit about plant, insect, and virus interactions. And so I thought it'd be best to start off with a little bit of history about plant virology. So the guy that we see over here is named Dmitry Ivanovsky, and he was a Russian scientist that was studying tobacco in 1892. And he was trying to figure out why the tobacco had this mosaic patterning on the leaves. And so he thought it was due to a bacteria. And so he took some infected sap from one of these plants and he ran it through a porcelain filter. And this filter was so fine that bacteria couldn't pass through it. And so he expected that the sap that he got when he rubbed it on a healthy plant would get no reaction. But instead, when he rubbed the sap on a healthy plant, he noticed that that healthy plant became infected. So you knew that it was something smaller than bacteria, but back then they didn't really have any idea of what it possibly could have been. So it wasn't until years later that they actually came up with the concept and idea of what a virus is. But he's considered to be a founder of virology in general. So, how do we name plant viruses? I think of plant viruses as being broken up into two parts. The first part is going to be the plant that this virus was first found on, and then we're going to follow that in its name with whatever symptoms it has that are the most prevalent. So, over here, this is all one virus. You can see we got this tomato. The tomato has some spotting, some like ring patterns. Then over here, the leaves look kind of wilted. They're dying. So this one's called tomato spotted wilt virus. This one over here, it's some type of bean plant, and we can see that it's got a really bad mottling pattern of the leaves. So it's probably not a surprise that this one's called bean pod mottle virus. So how are viruses transmitted? You might think that as soon as an insect bites into an infected plant, anything he eats from there on out is going to become infected. But that's not always the case because there's two things that can play a big role: acquisition time and latent time. Now, acquisition time is how long does it take for the insect to physically acquire that virus? So, how long does he have to be eating that plant before he's got the virus particles all around his mouth or in his mouth or inside of him? But latent time is how long can it take before that insect can spread that virus? How long before it becomes infectious? Because sometimes these viruses have to get into the gut or somewhere where they can replicate first. And so these two things on top of your mode of transmission is going to really make a difference for the virus when it comes to the ease of infection. So now we're moving on to the modes of transmission. I relate these modes of transmission to traveling, like ways of getting around. And so, starting with passive transmission, this is where the insect isn't actively involved. It's not really hurting anything to spread this virus or this pathogen, but there is transmission because of this insect. And so, let's say you're out on a picnic and this fly lands on some fecal matter nearby, and then that fly lands on your cheeseburger. Well, I mean, the fly wasn't like trying to passage this bacteria, but now when you eat that cheeseburger where that fly was, you're going to eat that bacteria. You're probably going to get sick. Another example would actually be pollen can transfer viruses. Some viruses like Nipah viruses and viruses can infect pollen. And if an insect was to roll around in the pollen and then fly to a healthy plant and pollinate it, it would probably be spreading that virus through the pollen as well. And so I rate passive transmission a hitchhiker when it comes to getting around. Up next, we got non-persistent transmission. With non-persistent transmission, that bug's going to eat that infected plant and it's going to get the virus particles inside or around its mouth parts, but just for a brief amount of time. So there's no latent period, and anything that it eats right away is basically going to be capable of becoming infected right away. And it can cause this infection while it's on its mouth parts for minutes to hours. So I gave it a Marty McFly on a skateboard because it's not ideal, but it's definitely better than walking. It's going to get you where you need to go, but it's not something that you want to do forever. Next up, we got semi-persistent transmission. Semi-persistent transmission is when the insect's going to consume the virions and it's going to carry that virus in its stomach or its gut tissues. And so there might be a short latent period, where the virus has to actually reach the stomach or gut tissues before it's capable of finding a way to spread the infection. And usually this is going to be capable of transmitting the virus for hours to days. So we got to step up, figure it's time for the Greyhound now. It's going to get you where you need to go. It's going to be better than skateboarding, but it's not a car of your own, is it? It's not something you really want to be in for too long. Okay, and moving on to our final type, we got persistent transmission, which has two types actually. So persistent transmission, not only are those virions inside the stomach or the gut tissues, they now have the ability to move to hemolymph, which would be the insect blood, or additional tissues like nerve tissues or muscle tissues. And so non-propagative persistent is when these virions are inside, they got all these nice tissues, but they're not replicating. So even though they're able to go, let's say, wherever they want in the insect, there's really only a finite amount of the virus. And once we're rid of it, we're not going to be having more. So I gave this a camper. It's luxury. It's really nice, but it could be better. How could it be better? Oh, persistent propagative. This is the golden ticket of being a virus. If you have this, you're basically set for life. Because if you're propagative persistent, you got access to not just the guts, maybe the hemolymph and other tissues. You're now replicating inside that insect as well. So it's kind of like a virus factory. And as long as that insect is going to be alive, the virus is going to keep replicating, which means that potentially that insect's going to be able to cause that infection all the way until it dies. So you might be thinking, well, I see a lot of bugs in my yard. Are all of these bugs equally capable of transmitting pathogens? Well, that's not always the case. In fact, a lot of these viruses and insects have really highly specific interactions that might as well be locks and keys when it comes to spreading these pathogens. And so one species of an insect may be capable of spreading one virus, but then only that virus. And an example I have here is the silverleaf whitefly can transmit tomato yellow leaf curl, but not tomato infectious sclerosis virus, despite these two being fairly similar. And so now I'm just going to go through the whole mode of infection for a virus. And the virus that I picked was tomato spotted wilt virus, or TSWV. This can be a really big problem if it happens in a field for a farmer. It can infect over 800 plant species. So all your big boys, tobaccos, tomatoes, crops, even flowers. And the symptoms that you're going to have with this disease, you're going to get some stunting, so the plant's not growing as good as it could, some wilting, it's not really doing too well, necrotic lesions of the leaves, some spots and circles, and it's going to be spread by this guy right here. If you don't know who this guy is a thrips, and it's always thrips. Even if it's just one of them, you're going to throw that S on the end. And thrips are in the order Thysanoptera. They got 6 legs, and they've got these fringed wings right here that are really just more like an appendage with hair on it, is what it looks like. And they don't use these to fly. They use these to ride throughout the air on air matrices or air currents, and they're not really meant to travel a whole lot with these wings that they have. And they're really, really small. I'm talking like if you look on your keyboard at your comma, that's probably generous for the size of an adult. So you can imagine you have a lot of these in your field capable of spreading the virus. It's a real nightmare. But how do they spread the virus? Well, they're very specific because thrips have 5 life stages if you don't include the egg: 2 larval, 2 pupal, and then the adult. So let's start off with this egg, and let's say that this egg is on a plant that's already infected with TSWV. That egg is going to hatch into a larva, and the larvae are going to start eating that infected plant. So these larvae are going to have the infected tissue in their gut. They can't spread it yet, but they have this infected tissue inside them. They're going to keep on progressing throughout their life cycle until they reach their pupa stage. At their pupa stage, they're going to drop off of the plants that they're on and go underground for metamorphosis. While they're metamorphosizing, something's going to happen inside their body that allows the virus that's been replicating inside their gut to come into contact with some salivary tubes. So that as an adult, and only as an adult, anything that he eats, I don't know why I said he, anything that the bug eats is going to salivate into the plant and spread the virus through the salivation. But only if he acquired it in these larval stages. If an adult eats a plant and it's not previously infected, it's going to get those virions in its stomach, but it's not going to be able to spread it through the saliva because it didn't have them for that metamorphosis. And again, if you refer to the previous slides, this would be propagative, persistent, because once the virus is inside, it's replicating inside the thrips. And I think I could show you this a little bit better with this picture I drew. Could use some work still, but so this is the pathway to infection, and you can ignore I wrote orthotospovirus here. This is the genus that tomato spotted wilt virus is in, so it's going to cover for this still. But the virions are going to go up through the esophagus, and this would still be in the larval stage. And then they're going to be swallowed into the midgut and into the stomach, where they're going to be replicating. So they replicate, you get a whole lot of the virions, and then that metamorphosis is going to happen where this tubular salivary gland is not directly maybe in contact with, but close enough for the virions to travel throughout the muscle tissues into this gland. So now they're in this gland where they reach more salivary glands, and they can leak out of those salivary glands into the salivary duct. So anything that thrips is going to eat afterwards, if he drools into it, it's going to have the virus inside of it. So I hope that was easy to understand. If you have any questions about anything, you can ask the moderators, or you could wait till the end and if you have something you want to ask with the mic. But until then, I'm going to turn it over to my friend Lewis, who's going to be talking about vector entomology. Thanks for your time.
Hi, everyone. Just bear with me. I have to share my screen and get my presentation up and running. Whoops, it's not the right one. All right. Again, my name is Lewis, and I am a graduate student in the Department of Entomology and Plant Pathology here at Auburn University. And today I'll be talking to you about Aedes aegypti and dengue fever. So this will be a brief overview of some insect vectors and a couple of different other human diseases. And here's the outline. As I said, we'll be covering human diseases vectored by transmitted by arthropods, and Aedes aegypti life cycle, and then the dengue virus infection in both the insect and human. And we'll end with control methods for Aedes aegypti. So before we get started, we need to know what a pathogen is. It is an infectious agent that can cause disease, can be viral, bacterial, fungal, or any other organism like Plasmodium, which causes malaria, which we'll discuss in the next slide. A vector is any agent that carries and transmits a pathogen to another living host. So what's different between vector and reservoir? A reservoir is an organism that is a host for pathogens in which the pathogen does not cause disease and is able to replicate inside of the host. So as mentioned in our previous lecture, there are some insects that can pick up viruses. They can be vectors, but they can't be reservoirs in that the virus that they pick up can't replicate inside of the insect cells. So malaria is a disease caused by Plasmodium, which is a single-celled eukaryote. The way it gets into the human is from an infected mosquito. So when the mosquito inserts its mouth part, the Plasmodium travels with the saliva into the bloodstream. So first, it has to travel to the liver, where it'll mature and it'll undergo asexual reproduction. Once it does that, it'll exit the liver cells, and then it'll go on to infect the blood cells. After which they form gametocytes, which can then be picked up by mosquitoes, and they can further their life cycle in mosquitoes, and then it just goes back and forth. Oh, and this is a widespread disease in tropical and subtropical regions. Most deaths are caused by the Plasmodium falciparum. That's all I wanted to say about that. Next, we'll talk about Chagas disease. This is another single eukaryote type disease, single-celled, and it is transmitted by the kissing bug. Those bugs in the subfamily Triatominae. I don't have a picture up here, but Rodnius is a species that can transmit this this organism. So the organism is called Trypanosoma cruzi. It is also known as American trypanosomiasis. So the early infections, it could be no to mild symptoms, fever, headache, or swollen lymph nodes. But as the disease progresses, usually there's no more pathology, but further down the line, you could develop heart disease. So the trypanosoma is not actually vectored through the bite. So it actually is vectored by the feces. So when an insect crawls on you, feces gets all over you, and then when it bites, that is a mechanical damage, allowing the agent to enter your cells. 6.2 million people are infected. This is in 2017, and mostly in Central and South America. The interesting thing about this organism is that it'll infect various cells, and then once they replicate a lot, they explode the cells. Next, we'll talk about the Rocky Mountain Spotted Fever. This is vectored by ticks, mostly those that belong in the subfamily Ixodidae. The disease is, the actual bacteria is called Rickettsia rickettsiae. And this is an interesting bacteria because it is maternally transmitted, and it is an obligate endosymbiont for the tick. Some symptoms include fever, headache, rash. So for this one, this is a vector and a reservoir, meaning that because the Rickettsia is endosymbiont, it lives with the tick, and it can be transmitted to people. And it enters your through bites, and it travels with the saliva when the insect is feeding on your blood. One more interesting thing about this is it requires 4 to 6 hours of attachment, meaning the tick has to be on you, attached and feeding for 4 to 6 hours before you can successfully acquire the disease. And then now we'll go into more specifically Aedes aegypti and the virus that it can vector. So for example, it can vector Zika, Mayaro, yellow fever, chikungunya, and then we'll discuss further dengue virus. So Aedes aegypti lives in tropical and subtropical regions, and it has spread to the US. The other Aedes mosquitoes is not popular, but well established in the States is the Aedes albopictus. Now that one is hardier, and it can withstand colder conditions than Aedes aegypti. So only the females feed on blood, and the blood meal is required for egg production. So we'll go into their life cycle. Oh, maybe not. Sorry, I don't have my slides in the right order. So the life cycle of Aedes aegypti, there's 4 life stages: the egg, larva, pupa, and the adult. So after a successful blood meal, the female would lay her eggs near water sources, and when conditions are conducive, the eggs will hatch into larvae, and then larvae feed on detritus, dead plant material. They can feed on fungi and other single-celled organisms in the water column. They will molt a couple times, and then they will turn into the pupa. And the interesting thing about the pupa is that it is fully motile, so it can still swim around. Another thing I forgot to mention is that they have this anal tube called a siphon, which they use to breathe. And another fact, for those of you that missed the Kahoot quiz, I asked, can both male and female mosquitoes feed on flower nectar? And the answer is yes. So a little bit about the mouth part. As you can see in this video, these things, these needle-like structures are called fascicles, and there are 6 of them that enter the cell, or no, that break your skin and enter. So here we have the maxilla. I'm just gonna go over this quick. It's kind of like more technical, but the maxilla is important for piercing the skin. Then we have the mandible, which is important for holding tissue. And then we have the labium, that remains outside. Then we have the hypopharynx and the labrum that work together. The labrum houses the sensory organs, so this is how it can, like, this is responsible for sensing blood vessels. This is like a homing device. And then the hypopharynx is right there, so it can, once the blood vessels are found, it can just pierce the blood vessel. So what is released is a saliva that has antihistamines and anticoagulants, so the blood can keep flowing, and the host doesn't feel the mosquito feeding on you. This is where the virus would enter your body, is through the saliva when it feeds. Whoops, I missed a slide. So dengue virus is a flaviviridae virus, and it is a plus sense RNA virus. So if we recall from the first lecture, this virus can directly undergo translation, meaning RNA to protein. So with that first step, the first protein that comes off is called a polyprotein. With host proteases, that gets chopped up into other pieces that can then form a mature ribozyme. So that viral proteins. So over 390 people are infected a year. It is a small virus, I think, because it only has 11,000 base pairs. And there are 3 structural proteins and 7 non-structural proteins. So the way it works is I think I have a figure somewhere that, yes, here. So the virus cycle is like the infected mosquito infects the human, infected human, and then the mosquito can pick it up from an infected human, and it's just a cycle. So these are closely evolved, because these Aedes aegypti, evolved near water sources that were near humans. So it made sense that there would be a closely evolved virus system. So, as I mentioned, the mosquito acquires the virus from infected humans, but also can come from contaminated water sources. This is the case when an infected human sheds viral particles and it enters water streams. This is like a common problem in countries that don't have water treatment, treated water. So infected mosquitoes, they have to, the virus has to incubate in them for about 4 to 7 days before they can infect another person. But as long as the mosquito is alive, it can transmit the virus. Viral replication is tightly controlled in mosquitoes. There are RNA interference pathways that keep it in check. So dengue in humans has a incubation period of 3 to 14 days, meaning it has to find a cell and start getting replicated. And there's a wide range of symptoms, and illnesses can last 3 to 7 days. And as I mentioned earlier, when people start shedding the virus, this is called viremia, virus in the blood. So this is the overall mechanism for viral entry in both insect and human cells. So the virus has to bind to a toll receptor, and then it triggers toll receptor mediated endocytosis, meaning the cell will form this cavity and eventually it'll just close, and then inside is the virus within a double bound membrane bound type organelle called endosome. And then the pH changes, which causes a conformational change in the coat protein, and then it turns into spikes, which then pierces the endosome, allowing the release of the genetic material. So since it is mentioned earlier, it is plus sense RNA strand, it can directly go translation, which can form the needed viral proteins for viral replication. So the assembly of the virus happens near the ER, as you can see here, and it gets packaged and it has to go through several membranes of the Golgi in order to become a mature virion. So the reason it has to go through the Golgi body is the change of pH, because you don't want the virus to, have its coat protein undergo conformational changes. If it did that, it'll just exit the membrane and infect the cell again, and you don't want that. The virus wants to leave. So after it leaves the Golgi, normal cellular mechanisms, signals, take this out of the cell. So that's exocytosis. So how is this currently being managed? So for you, as you going out in the wild, you want to protect yourself. You want to first, avoid getting bitten. So people, there have been various chemicals developed to repel these mosquitoes, such as DEET. Wear protective clothing. Whoops. And some people in other countries have nettings that they hang from the ceiling, so that prevents mosquitoes from getting in. So I think that's the most important management, is prevention. But there have been other programs where, like in Singapore, they release Aedes aegypti, either sterile or Wolbachia infected. What this does is, with the sterile males, that reduces the population of mosquitoes. So if you reduce the population, then you lessen the risk of the virus infecting other people. With the release of Wolbachia infected female mosquitoes, this bacteria confers resistance to viral infections. So there's been lots of studies that have shown when an infected mosquito gets infected with virus, the virus usually doesn't make it out of the mosquito. So this is a good management option if you didn't want to release sterile males. I feel like the Wolbachia infected is a bit more organic than releasing a transgenic sterile male, to each their own. So, and then there's also biocontrol agents such as entomopathic fungi and bacteria. As you can see in this picture, this guy has been parasitized by fungus. And here's one of these release programs. So people just get these mosquitoes and just release them. And currently in our lab, we're working on a Wolbachia system to be used in mosquitoes. So we're working on that. Again, there's people developing new protective clothing to prevent biting. There's all sorts of control management techniques. And with that, thank you for coming, and I'll take any questions.